The human body consists of an organ system which keeps us alive, such as the lungs, which oxygenate the blood, and the heart, which pumps it around to the rest of the body. In the event of traumatic injury or disease, an organ or multiple may fail and require a life-saving organ transplant. Scientists have developed external systems to keep donor organs alive while awaiting transplant. This increases the number of active organs that are available for patients on the wait list and potentially allows doctors to treat damaged organs and make them suitable for transplant. These external methods to keep donor organs alive are called ex vivo machine perfusion systems. Ex vivo, meaning outside of the body, and machine perfusion relating to the passage of fluid through tissue aided by mechanical pumping. The perfusion system takes over essential tasks that keep organs alive, such as providing oxygen and pumping a nutrient-rich fluid through the vessels. The advantage of this system is that it can allow for functional recovery of the organ, observation of many viability parameters, and increased flexibility for the timing of the transplant surgery. The alternative to machine perfusion is storage on ice. While depressing the metabolic system increases preservation time, the static state of the organ does not allow for functional tests or treatment. Current methods typically begin treatment with static cold storage and follow with machine perfusion to gain the benefits of both techniques. In our lab, we perfuse livers, hearts, kidneys, pancreas, and vascularized composite allografts. Here we have a machine perfusion system built for large organ systems. And here is one for small organ systems. The components are as follows: a mechanical pump to drive the fluid through the circuit, an oxygenator to increase diffusion of oxygen, a bubble trap to remove air bubbles from the perfusate. Next we have a basin to hold the organ, and a pressure sensor to monitor resistance of the organ's vessels, a water bath for temperature control, and tubing to connect all of the pieces. Perfusate is the fluid flowing through the system which carries essential nutrients, like amino acids and sugars. It has buffers to maintain a physiological pH, and components to maintain osmotic pressure and viscosity of the solution to ensure full patency of the vessels and reduce edema. For very large organs, the level of oxygen diffused into the perfusate is insufficient to supply the full quota of oxygen required. Thus, it can be necessary to have a dedicated oxygen carrier, like red blood cells, which have a very high affinity for oxygen and increase the available oxygen in the perfusate. Use of an oxygen carrier will guarantee full oxygenation of the periphery of the organ. However, using red blood cells introduces extra variables into the system, such as age of donated blood cells, immune system recognition, and potential clotting. Here, a closed-loop system is shown. The perfusate is circulated continuously. Perfusate leaves the pump to travel directly to the oxygenator. Next is the bubble trap, which directly precedes the cannula to the organ to remove any air bubbles which can cause an embolism in the blood vessels. The perfusate then enters the organ through a cannulated vessel and can exit through a second cannulated vessel or freely into the basin. The flow set by the pump can be continuous or pulsatile. For venous perfusion systems, such as liver perfusions through the portal vein, this pulsatility must be reduced to avoid damage to the veins, since they don't have a muscular wall like arteries. This can be done by adding an air-filled syringe to the bubble trap to buffer out abrupt pressure changes. Tubing comes in a large set of gauge sizes. For example, we would use a large tube for human liver to accommodate for higher fluid mass transfer. Our pump is compatible with a range of thread sizes, which we change accordingly. The flow rate can be precisely controlled to affect the pressure. Decreasing flow rate can work to decrease pressure within the organ. To reduce heat loss and the total volume contained in the system, the total length of the tubing should be kept to a minimum. From the pump, the perfusate travels to the oxygenator. The oxygenator is linked to a tank with atmospheric gas, which delivers oxygen to the chamber and can be controlled to deliver either higher or lower amounts of gas. The oxygen diffuses into the perfusate through the semi-permeable membrane. There are different types of oxygenators. Here we are using a coiled silicone oxygenator, and here is a hollow fiber oxygenator, which has bundled fibers to increase the rate of oxygen diffusion. In the bubble trap, bubbles rise to the top and air is forced out of the bottom. As a bubble can damage or even kill any organ, this is an essential part of the machine perfusion system. We ensure that the bubble trap is at least half full to allow for space for air to rise without fully emptying the bubble trap and forcing air into the organ. All of the glass components of the system are double-walled to run temperature-controlled water through the system. For normal thermic machine perfusions, we set the temperature to 37 degrees Celsius. This is to mimic normal body temperature so that the metabolism is maintained at physiological rates. To reduce the metabolism, the chiller can be set to different temperatures, such as 21 degrees Celsius or even lower. The pressure experienced within the organ is one of the most important measurements and in most cases determines the flow rate, as pressure and flow are directly related. Pressure corresponds to resistance experienced within the blood vessels. Too high pressure can cause fluid to leak into the tissue, also known as edema, or it can cause endothelial damage. Too low pressure can lead to insufficient perfusion at the edges of the organ. To accurately measure pressure in the organ, the pressure sensor must be placed at the same height of the organ. First, we must calibrate or zero the pressure monitor. To do this, the gauge will be opened and the sensor will fill with perfusate. The flow is paused until the dripping stops and the movement of fluid comes to a standstill. We press the zero or calibration button and the gauge is closed. From this moment on, it is important not to move the position of the sensor and cannula in order to secure accurate readings. Keep in mind that the pressure monitor reads the pressure in the system plus the pressure in the organ. Before each perfusion, we record the base pressure over the range of flow rates that we plan to use. For example, in human liver perfusions, we record the pressure of fluid flow from 50 to 800 mL per minute in increments of 10 mL per minute. While taking system pressures, make sure that the syringe on top of the bubble trap is depressed, so that air cannot escape into the syringe, ensuring accurate readings, and make sure that a cannula is attached. Once the organ is attached, we can read the pressure of the entire system and subtract the base pressures to obtain the pressure within the organ. In this figure, we can see the resistance in the vessels decreasing over time. We adjust the flow rates incrementally to maintain the ideal pressure range, which varies between organs. When connecting the organ, increase the flow slightly and point both the cannula and the inflow tube in upward directions to avoid air bubbles from entering the system. Make sure that the cannula is filled with liquid, and as liquid slightly overflows from both tubes, carefully slide the cannula onto the inflow tube in one smooth movement. Take care not to manipulate the cannula too much, as this can cause it to dislodge from the organ. Finally, make sure that the cannula is not twisted and uninhibited flow of perfusate is allowed into the organ. Viability of the organ can be assessed through different assays. Samples of perfusate can be obtained at the inflow and outflow vessels, so we can monitor changes within the organ. When taking the sample, care must be taken not to retract the sampling syringe too quickly, as this will cause the flow into the organ to stop and air to enter the system, as shown right here. Checking key levels of parameters allows for real-time interference, such as adding additional pH buffers or increasing the oxygen flow. We can also look at different markers for metabolic function and damage. Metabolic markers include oxygen consumption and lactate levels. Damage markers include ALT and AST in livers and troponin in hearts. Weight of the organ before and after perfusion is used to track edema. The future of machine perfusion will allow for advanced functional tests to guarantee the highest quality of organs for transplant, extending the preservation time of organs and treatment options like cell therapy and genetic engineering.